Hybridoma technology is a method for producing large numbers of identical antibodies, also called monoclonal antibodies. This process starts by injecting a mouse, or other mammal, with an antigen that provokes an immune response, a type of white blood cell. The B cell that produces antibodies that bind to the antigen are then harvested from the mouse. These isolated B cells are in turn fused with immortal B cell cancer cells, a myeloma, to produce a hybrid cell line called a hybridoma, which has both the antibody-producing ability of the B cell and the exaggerated longevity and reproductivity of the myeloma. The hybridomas can be grown in culture, each culture starting with one viable hybridoma cell, producing cultures each of which consists of genetically identical hybridomas which produce one antibody per culture. Monoclonal, rather than mixtures of different antibodies. Polyclonal. The myeloma cell line that is used in this process is selected for its ability to grow in tissue culture and for an absence of antibody synthesis, in contrast to polyclonal antibodies, which are mixtures of many different antibody molecules. The monoclonal antibodies produced by each hybridoma line are all chemically identical. The production of monoclonal antibodies was invented by Cesar Milstein and George J. F. Kohler in 1975. They shared the Nobel Prize of 1984 for medicine and physiology with Niels Kajun, who made other contributions to immunology. The term hybridoma was coined by Leonard Herzenberg during his sabbatical in Cesar Milstein's laboratory in 1976-1977. Method: laboratory animals, mammals, e.g., mice, are first exposed to the antigen that an antibody is to be generated against. Usually this is done by a series of injections of the antigen in question over the course of several weeks. These injections are typically followed by the use of in vivo electroporation, which significantly enhances the immune response. Once splenocytes are isolated from the mammal's spleen, the B cells are fused with immortalized myeloma cells. The fusion of the B cells with myeloma cells can be done using electrofusion. Electrofusion causes the B cells and myeloma cells to align and fuse with the application of an electric field. Alternatively, the B cells and myelomas can be made to fuse by chemical protocols, most often using polyethylene glycol. The myeloma cells are selected beforehand to ensure they are not secreting antibody themselves and that they lack the hypoxanthine guanine phosphoribosyl transferase (HGPRT), John, making them sensitive to the HAP medium. See below. Fused cells are incubated in HAP medium, hypoxanthine-aminopterin thymidine medium, for roughly 10 to 14 days. Aminopterin blocks the pathway that allows for nucleotide synthesis. Hence, unfused myeloma cells die, as they cannot produce nucleotides by the de novo or salvage pathways because they lack HGPRT. Removal of the unfused myeloma cells is necessary because they have the potential to outgrow other cells, especially weakly established hybridomas. Unfused B cells die as they have a short lifespan. In this way, only the B cell myeloma hybrids survive, since the HGPRT gene coming from the B cells is functional. These cells produce antibodies, a property of B cells, and are immortal, a property of myeloma cells. The incubated medium is then diluted into multi-well plates to such an extent that each well contains only one cell. Since the antibodies in a well are produced by the same B cell, they will be directed towards the same epitope, and are thus monoclonal antibodies. The next stage is a rapid primary screening process, which identifies and selects only those hybridomas that produce antibodies of appropriate specificity. The first screening technique used is called ELISA. The hybridoma culture supernatant, secondary enzyme labeled conjugate, and chromogenic substrate are then incubated, and the formation of a colored product indicates a positive hybridoma. Alternatively, immunocytochemical screening can also be used. The B cell that produces the desired antibodies can be cloned to produce many identical daughter clones. Supplemental media containing interleukin-6, such as briclone, are essential for this step. Once a hybridoma colony is established, it will continually grow in culture medium like RPMI1640, with antibiotics and fetal bovine serum, and produce antibodies. Multi-well plates are used initially to grow the hybridomas, and after selection, are changed to larger tissue culture flasks. This maintains the well-being of the hybridomas and provides enough cells for cryopreservation and supernatant for subsequent investigations. The culture supernatant can yield 1 to 60 micrograms per milliliter of monoclonal antibody, which is maintained at -20 degrees Celsius or lower until required. By using culture supernatant or a purified immunoglobulin preparation, further analysis of a potential monoclonal antibody-producing hybridoma can be made in terms of reactivity, specificity, and cross-reactivity. Applications. The use of monoclonal antibodies is numerous and includes the prevention, diagnosis, and treatment of disease. For example, monoclonal antibodies can distinguish subsets of B cells and T cells, which is helpful in identifying different types of leukemias. In addition, specific monoclonal antibodies have been used to define cell surface markers on white blood cells and other cell types. This led to the cluster of differentiation series of markers. These are often referred to as CD markers and define several hundred different cell surface components of cells, each specified by binding of a particular monoclonal antibody. Such antibodies are extremely useful for fluorescence-activated cell sorting, the specific isolation of particular types of cells. In diagnostic histopathology, with the help of monoclonal antibodies, tissues and organs can be classified based on their expression of certain defined markers, which reflect tissue or cellular genesis. Prostate-specific antigen, placental alkaline phosphatase, human chorionic gonadotrophin, alpha-fetoprotein and others are organ-associated antigens in the production of monoclonal antibodies against these antigens helps in determining the nature of a primary tumor. Monoclonal antibodies are especially useful in distinguishing morphologically similar lesions, like pleural and peritoneal mesothelioma, adenocarcinoma, and in the determination of the organ or tissue origin of undifferentiated metastases. Selected monoclonal antibodies help in the detection of occult metastases, cancer of unknown primary origin, by immunocytological analysis of bone marrow, other tissue aspirates, as well as lymph nodes and other tissues, and can have increased sensitivity over normal. Histopathological staining. One study performed a sensitive immunohistochemical assay on bone marrow aspirates of 20 patients with localized prostate cancer. 3 monoclonal antibodies, T16, C26, and R1, capable of recognizing membrane and cytoskeletal antigens expressed by epithelial cells to detect tumor cells, were used in the assay. Bone marrow aspirates of 22% of patients with localized prostate cancer. Stage B, 0/5, Stage C, 2/4, and 36% patients with metastatic prostate cancer. Stage D1, 0/7 patients. Stage D2, 4/4 patients, had antigen-positive cells in their bone marrow. It was concluded that immunohistochemical staining of bone marrow aspirates are very useful to detect occult bone marrow metastases in patients with apparently localized prostate cancer. Although immunocytochemistry using tumor-associated monoclonal antibodies has led to an improved ability to detect occult breast cancer cells in bone marrow aspirates and peripheral blood, further development of this method is necessary before it can be used routinely. One major drawback of immunocytochemistry is that only tumor-associated and not tumor-specific monoclonal antibodies are used, and as a result, some cross-reaction with normal cells can occur. In order to effectively stage breast cancer and assess the efficacy of purging regimens prior to autologous stem cell infusion, it is important to detect even small quantities of breast cancer cells. Immunohistochemical methods are ideal for this purpose because they are simple, sensitive, and quite specific. Franklin et al. Performed a sensitive immunocytochemical assay by using a combination of 4 monoclonal antibodies, 260F9, 520C9, 317G5 and BRA3, against tumor cell surface glycoproteins to identify breast tumor cells in bone marrow and peripheral blood. They concluded from the results that immunocytochemical staining of bone marrow and peripheral blood is a sensitive and simple way to detect and quantify breast cancer cells. One of the main reasons for metastatic relapse in patients with solid tumors is the early dissemination of malignant cells. The use of monoclonal antibodies, mAbs, specific for cytokeratins, can identify disseminated individual epithelial tumor cells in the bone marrow. One study reports on having developed an immunocytochemical procedure for simultaneous labeling of cytokeratin component No18, CK18, and prostate-specific antigen, PSA. This would help in the further characterization of disseminated individual epithelial tumor cells in patients with prostate cancer. The 12 control aspirates from patients with benign prostatic hypertrophy showed negative staining, which further supports the specificity of CK18 in detecting epithelial tumor cells in bone marrow. In most cases of malignant disease complicated by a fusion, neoplastic cells can be easily recognized. However, in some cases, malignant cells are not so easily seen or their presence is too doubtful to call it a positive report. The use of immunocytochemical techniques increases diagnostic accuracy in these cases. Gush, Mason and Spriggs analyzed 53 samples of pleural or peritoneal fluid from 41 patients with malignant disease. Conventional cytological examination had not revealed any neoplastic cells. 3 monoclonal antibodies, anti-CEA, CA1 and HMFG2, were used to search for malignant cells. Immunocytochemical labeling was performed on unstained smears, which had been stored at -20 degrees Celsius up to 18 months. 12 of the 41 cases in which immunocytochemical staining was performed revealed malignant cells. The result represented an increase in diagnostic accuracy of approximately 20%. The study concluded that in patients with suspected malignant disease, immunocytochemical labeling should be used routinely in the examination of cytologically negative samples and has important implications with respect to patient management. Another application of immunocytochemical staining is for the detection of 2 antigens in the same smear. Double staining with light chain antibodies and with T and B cell markers can indicate the neoplastic origin of a lymphoma. One study has reported the isolation of a hybridoma cell line, clone 1E10, which produces a monoclonal antibody, IgM, K isotype. This monoclonal antibody shows specific immunocytochemical staining of nucleoli. Tissues and tumors can be classified based on their expression of certain markers. With the help of monoclonal antibodies, they help in distinguishing morphologically similar lesions and in determining the organ or tissue origin of undifferentiated metastases. Immunocytological analysis of bone marrow, tissue aspirates, lymph nodes, etc. with selected monoclonal antibodies help in the detection of occult metastases. Monoclonal antibodies increase the sensitivity in detecting even small quantities of invasive or metastatic cells. Monoclonal antibodies, MABs, specific for cytokeratins, can detect disseminated individual epithelial tumor cells in the bone marrow. Hybridomas at the U.S. National Library of Medicine. Medical subject headings: MESH, Hybridoma Technology, Understanding Cancer Series, The Immune System, National Cancer Institute. Archived from the original on 5 October 2014. Hybridoma cell culture.